Welcome back to Big Ideas at Microscale. Today we have a master's student from McGill University. And I think during our preliminary conversations with him, he's dove deep into sustainable manufacturing. And I think it would be an extremely interesting conversation with him. My name is Hemdeep, and my co-host is...
Robin, that's me.
Great. And so today we're going to have Alex join us, and here he is. I'm sure that he'll have a very interesting conversation with us, and I know I've got a ton of questions to ask him as well. Welcome, Alex, to our little podcast here. How are you doing?
Well, thank you for having me here. I'm very happy to be able to come exchange ideas with you all.
I guess we can quickly dive deep into, who you are, the lab that you work at. How did you get there, sort of your pathway to where you are now?
Excellent. Yeah, absolutely. So I'm Alex. I'm from Montreal. I grew up all my life in Montreal. I studied chemical engineering at McGill University, did my undergrad there, and then joined the Cellular Microenvironment Lab in 2023, so right after I graduated from my undergrad in chemical engineering. And so this is a little bit about where I am right now. So the lab is mainly, it's kind of an interesting lab where we have really different projects going all around. So people, for example, working on type 1 diabetes, breast cancer, for example, air-liquid interface with a differentiation of epithelial cell to go to lung, and really a lot of different things. And mainly all of this lab kind of studies this, the mechanobiology around all the differentiation of those cells. But for me, I'm kind of a different project, I'd say. It came from a project that I did in my class with Chris, that Chris was teaching, so my supervisor. And one of our projects that we had in class was to make this sustainable mycelium composite that I was very intrigued with, since the way he was framing it was, if you put, for example, the root-like part of the mushroom on some wood kind of waste byproduct, it would form this material that had the same properties as polystyrene, but would be fully biodegradable. And we had to do one of those projects. And I was like, oh, this is super interesting to do as part of a class. And when I was looking towards my master's, I was like, oh, this could be a very interesting project, since I'm kind of always been towards a little bit more sustainability, especially in the material with chemical engineering. So I pitched in the project. I was like, hey, could that be something that could be interesting for you? I know it's not specifically what you're doing in the lab, but this could be something very interesting. And he was like, yeah, why not? Let's give it a try. And well, here I am now.
So this is one of those unique moments where you take a classroom lesson and you find a actual possible commercial endeavor, and you're solving a solution. Was this your first time that you ever saw that connection between classroom lesson and a real world application?
I'd say so. With McGill, and especially in chemical engineering, we're very theoretical. We're very looking at even we don't really even see pumps, because everything is very like theoretical. So when we are in the lab building those sort of prototypes and devices, I was like, well, this is something that's very interesting, and that's kind of in my values of more sustainability. So I was like, okay, this is very, very interesting. I think this could be a very fun thing to see and develop further along.
Nice.
Is that normal? Like when you join a lab, for example, from what I understood, the PI is usually the one who kind of assigns projects. But in this case, it's the opposite, where you approach Chris more as yourself, and you're like, hey, I want to do this. And, he he was nice enough to be like, yeah, let's go for it.
So I'd say it kind of depends, as our lab also is quite different. There's really a lot of projects that are coming. And when I sent him the email, at first he was like, I really like that it's you who brought the project, because typically that means that students are more motivated. They're not coming just for a PI or just for the overall lab in itself that has a good reputation in all this. They're coming because they are motivated and want to do this. And I think this is kind of I'd say a little bit more special, a little bit different in terms of the experience of the masters as well. since I've heard also in other labs where it's very very by the book, so really it's very stringent, it's very we are a lab of this, we're only doing this, and this is this aspect that you're going to be looking into, whereas for us it's let's take different directions, like let's take some risks, and that's what I like about this lab as well.
So now in the lab itself, it sounds like if you have a number of disciplines doing their own research, there seems to be a lot of collaboration that needs to be in place, does it not? And so how many, for your project, obviously you're heading it, are you reaching out to a number of people within the lab in order to facilitate the work that you do?
So I'd say yes, because some of the different aspects, for example, that we've developed either with 3D printing or with different, for example, agarose mold, this has already been done in our lab. So some people have already worked a little bit, but even though your project is a little different, I'm able to get some of the ideas that have been developed a little bit by others and then use that into my research and take some of this. So really everyone is collaborating together. And also, sometimes we do see some big, I'd say, overlap between, for example, cells, for example, and, for example, in our case, mycelium. So there's some ideas that are sometimes coming back. And since people, for example, some postdocs, some PhD students that have different experience from different labs and different backgrounds, we're all kind of able to come in together and share ideas, even though some of the projects are kind of different.
Amazing. So let's talk a bit about more your research project specifically. Like, tell us about micromaterials. What are they? Like, what its purpose? Just so the audience have a better idea.
Absolutely. So, if I could summarize it a little bit in like the big line, so we're taking the root-like part of the mushroom. So some scientists, they discovered that this could be made. So we take the root-like part of the mushroom called the mycelium. So it's not like kind of the fruit part of the mushroom that you buy at the grocery store that you put in your soup. It's really the part that is underground. So we take this and we put it into sawdust or straw, for example. So kind of this, I'd say, this substrate, and we put it on there, and then slowly the mycelium is gonna start growing and very fully colonize this substrate to form this kind of brick that has similar properties, so especially similar density, but also similar heat insulation properties, such that it makes a similar properties such as styrofoam or even polyurethanes, but it degrades in 45 days in your backyard comparatively to 500 years for conventional styrofoam. And the neat part about this is that it's also self-regenerative. So as long as it's alive, we can take a piece of this already colonized mycelium, we take it, and then we put it into our substrate, and then slowly it's going to restart and start growing back again to fully form this new colonized material. And this is a very interesting application, especially for packaging, because currently when you receive, for example, a packaging, there's always going to be tons of styrofoam, which typically will just end up in the waste basically immediately. You receive, for example, a new appliance, or for example, we receive a new fridge in the lab, came with lots and lots and lots of styrofoam, and then when we receive that, it immediately goes to waste. And this takes approximately 500 years to degrade. But if, for example, we were to replace it with those sort of materials that can be functional replacement for the plastic, well, we can put it into our backyard and basically would degrade in 45 days.
What does the process look like for that? Do you just kind of dig a hole in your garden and you just put it in there and then you just, dig it back up? That's it? There's nothing else you would have to do?
Yeah, basically you would just, for example, it's fully compostable. You would just put it in your backyard and that basically would be it. It's all made of natural material. So mushroom and sawdust. So it would degrade. It would take the water, and then just the other parts of the soil would just start degrading it.
And the psyllium itself, does it need to be from a specific type of fungi, or it could be? Do you guys grow it at the lab? How does that work?
Yeah, so we need a specific. So within So in the fungus ring, I'd say there's a lot of different type of fungus, but we are using one specific branch, which is called the white rod fungi, which that's the type of fungi that degrades the wood, for example. It's going to make, that's the ones that you see, for example, the forest, that is going to degrade the wood. And this is what we need. They need to be able to degrade the wood or the straw. And this is what we need in our case. So typically what we're going to be using is, in our lab we use Ganoderma lucidum, which is one of the two main strains that are being used. Otherwise, it's the oyster mushroom as well that we can use as well, that can be used. And also different strands are going to form a different, I'd say, a mat of mushroom as well. So for example, Ganoderma lucidum, the one that we use, form this very rigid mycelium mat, whereas the other one, the oyster mushroom, is going to form kind of this more fluffy type of mat. So even within the white rod mushroom, there's still some differences, but both of them can be used to make this packaging material.
So you just indicated that there was a link between the type of mushroom that you use and then the actual composite that you can connect it with. Effectively, your research that you're doing could be an idea. It could help people have new ideas of new combinations of mushroom with available, raw material, in this case. Correct?
So I would say my research is not to do combination. My research is more about how can we make those materials faster. Okay. So how can we, for example, make those larger scale materials faster, but with different type of, for example, mushroom strains, we can also do different type of materials as well. So we don't have to stay, for example, with the packaging. This is one very great example, and this is the one that's the most important for me. So this is what I want to try to get to, but also it can help make larger material that are for different applications. So for example, there's been some development of this type of material for example, leather-like materials. So for example, they've made, for example, coats that are made of mycelium leather. And now they're trying to do some woven fabric as well with this sort of mycelium material. So this is some very interesting aspect as well, that we can start to do some clothing as well from it. But one of our collaborators from UBC, Nicholas Lin, who's a postdoc in the architecture department, they're trying to make those very big scale buildings and overall, buildings, I'd say, for mycelium. They developed this kind of toilet that's supposed to replace the chemical toilets made of mushroom. And the very interesting part about this is that when they tried it, they saw that there was not this characteristic smell of chemical toilet, which was a very big surprise. So it was the mycelium overall material, even though the mycelium is completely dead, so it's just the overall, the remaining structure, it did not have the smell of the chemical toilet. So it was very interesting to see that as well.
Wow.
So the entire toilet was made out of mycelium? Like, I guess. Sorry, I'm just trying to wrap my head around that, what that looks like.
Yeah, me too. It's a very interesting thing to think about. But yeah, the whole toilet was made of like a substrate that has been colonized by mycelium. So then again, also I forgot to mention, but when we make those material for packaging, we basically autoclave the mycelium. So we basically pass it into the oven to make sure it's fully cooked and killed. So this way, the mycelium won't grow into your package. Exactly. So it won't grow on your package, and likewise, for example, it won't grow on the toilet. So that's some of the different, I'd say, horizons that we can get with this sort of new materials.
So I guess to some degree, it's quite waterproof as well in that case.
So that's kind of I'd say yes and no. There's a lot of, that's what makes it biodegradable, is that it can take up the water. And the reason why polymers and plastics are not biodegradable is because they're very not waterproof. So in our case, it is not that waterproof, which makes it biodegradable. But in case of the toilet, for example, where it is not that waterproof as well. But in their case, I think it might be waterproof just enough to be able to sustain a long period of time. I would need to get more details to to see how much it can withstand, for example. But it seemed to be good enough to withstand. I think they're doing a demonstration, I think, in September at UBC.
Interesting. I'm sure they figured it out, because I don't think anyone wants a disintegrating toilet while they're
exactly.
while it's in use.
Yeah, it won't be instantaneous, but over time, for example, it's slowly going to degrade. Yeah.
So degradation time was one of the properties that you kind of compare between micromaterials and polystyrene. Like, what are some of the other properties that you look at?
So for us, we're not like doing characteristics comparison in itself. We're really trying to speed up the growing process. But in the different papers that we've seen is, well, the cost is much lower since it's self-regenerative. And also, we don't need a lot of big instrumentation, for example. We don't need a lot of heating. We just basically let it grow. We can have it, for example, in the incubator at 30 degrees. So this also makes it carbon negative, or even carbon neutral, even carbon negative. So what I mean by this is, typically when we make polymers, polymers are typically made of oil, and the overall process consumes a lot of energy, a lot of electricity, and overall emits CO2 in the atmosphere. Whereas here it's a little different, where we basically almost capture CO2 to make those material, or it's very, very low CO2 emission. So there's a very big difference between this, and that's really one of the big characteristics between this and the overall plastic polymers. But then also the overall, the density, the insulation properties, but the main problem that we see and that we are trying to solve right here is the production time. Since plastics that take like minutes to hours to make, it's very not that long, whereas for those mycelium polymers, it take days to weeks to make. So it really takes a lot of time to be able to make this, and the longer it is, the longer it needs to be on your shelf, and the longer it is that the production time overall, so it costs more money, and there's more risk of contamination. So really our goal is to try to speed up this production process.
Okay, so let's kind of unpack that. I'm aware there's like generally 4 stages when you're making micromaterials based off like our previous conversation. You have the substrate integration and seeding, the mycelium colonizing, removing from the mold, and then the autoclave that you were just talking about, where you sterilize and devices, or the packaging is ready for use. So looking at that kind of process, which part of it is causing the lengthy production time?
Yeah, so it's really a colonization of the overall material, because the first step, as you mentioned, which is basically just, for example, we have our mold. That is the reason why it's very interesting, is because we can basically make any packaging out of it. As long as you have the right mold, we put, for example, the sawdust into that mold that will make the packaging that's, for example, perfect. For example, I don't know, a candle, for example, you want to send to somebody else. We're going to put it into this packaging. Then we put our mycelium on top of it. And this doesn't take very long. We make sure everything is sterile. And then we'll let it grow. And that's really the long part that we need to take 5 to 20 days or even longer, depending on the substrate that you're using or the quantity of mycelium that you put in. Then it takes a long time to be able to grow. And when it's fully colonized, we just simply take it out of the mold, we autoclave it. So overall, it's really the long part is really only the colonization time. So for the mycelium to really be able to fully cover the entirety of the substrate, this is really the long part of the process.
So if we were just to sort of take one small step backwards and sort of go through the actual work that you did, because I think the work you did wasn't on a 3-dimensional mold, but actually effectively on a 2D plate, correct? Yes. What did that work look like, and how did you, what was the challenges that you sort of faced, and what kind of optimizations did you guys find?
Yeah, so the first kind of question that we had to be able to see, okay, what could be the reason why it's causing this lengthy process, is the first thing that we thought was, is it the size? So I don't know if you have plants at home, But if you have ever tried to split your plants and to start a new, for example, colonization of your plants, for example, you have a couple orchid plants, or
Yeah, when you're
if you
propagating the plant.
exactly, if you want to
Yeah.
try to propagate your plants, I don't know if you've seen, but if the new plant that you're taking is too small, it's going to take some time to be able to grow back. And if it's too big, it also will take a longer time comparatively to when it's like just in the right size. It's really able to grow well. And I try to use this, I try to use this kind of idea for the mycelium. So we're like, is there an optimal size for the mycelium that we can split into and put it um, into those like 2D pattern agar plate, I'd say, and see if that makes a difference in the overall colonization. So what we did is we tested with three different sizes of plugs. So basically, we used biopsy punches, which is basically like fancy cookie cutters of 3 millimeter, 2 millimeter, and 1 millimeter to make these little cookies of agar mycelium that we put at different distances from each other. And what we saw is that, well, first, if they were further away from each other, it kind of resulted in this patchy uh, overall colonization, which is not optimal, which is not what we want to see when we have a full material. We want it to really be homogeneous, which is
Sorry to
yes.
pause you there. Do you know what the distance was between the mycelium cookies, as you call
Yeah,
it?
it was a couple millimeters that they were apart. So, for example, the closest that they were together was approximately 2 to 3 millimeters, and the further away was approximately 6 millimeters, approximately
Okay.
between the different cookies. And then from there, we saw that when they were a little bit further away from each other, it resulted in this patchy growth, which is not what we saw when they were a little bit closer together. That was our first observation, but also is that we made a defined area of the overall agar, which was approximately 34 millimeter overall, so basically the size of a well plate. And we saw that there was not a big difference between the plugs that were 1 millimeter and the plugs that were 3 millimeters. And that's when we were like, oh, this is very interesting, because uh, 18 we had 18 on those little 34 millimeter little discs, 18 plugs at different distances from each other. The smaller ones, they covered the overall same area, the overall 34 uh, uh, millimeters in diameter, at the same time as the 3 millimeters, which was very interesting to us since there's not the same amount of initial biomass. And this is where like, oh, so the size do seem to have an impact on the overall colonization. Since if, for example, we would apply this to a larger surface at equal biomass, the 1 millimeter plugs, which would be a lot times more, so 9 times more if you do the math in terms of the circle area, it would cover a lot faster and a lot more than at equal biomass, the 3 millimeter plugs. So if, for example, it was smaller and more dispersed, we would be able to colonize much faster than if it was bigger particles, well, fewer bigger particles. This is kind of what we saw with this 2D essay at the beginning.
So was that a function of not the biomass, but actually the surface area of each of your plugs and the contact point to the actual wood chip in this case?
So for here, we just did it on agar, so there was not
On agar,
any
sorry,
wood on this. So we think it's also it needs to have this balance between the nutrient and the overall like surface area, but also I think there's probably some sort of inhibition in between the mycelium at the beginning, which causes it to have some problems being able to fully expand and result in this patchy growth.
Okay.
Even though that when it recognized that it's the same mycelium, it's able to fuse and really form one. So this is how we're able to obtain this one full block at the beginning, is even though there's many, for example, different pieces of mycelium, when they recognize that it's the same exact DNA. They're going to fuse and form and become one overall mycelium. So this is what we're seeing, but it seems that even though they are further away from each other, they're not able to kind of fuse properly and form uniform coverage, whereas when they are closer together, they are able to fuse well and form this uniform coverage.
So now that you had a pretty good idea of like the mycelium plug sizes and the distance between them, how did you actually go about finding and narrowing down to get the optimal size, or if you even managed to get the optimal size?
Well, that's a very good question. So unfortunately, we don't have cookie cutters that are going below 1 millimeters. We don't have biop sponges that are going below, and it would be very difficult, even if we had to be able to take those little, very very tiny plugs and put them at different distances to each other. So we had to change a little bit our approach, and instead we decided to go with microfluidics. So what we did is we took a little piece of mycelium, put it on the exterior of a microfluidic chip, and we let the mycelium be able to colonize little channels inside PDMS microfluidic chip. So what we had is just a very basic design that had channels that were 100 micron tall by 100 micron wide, and they were 8 millimeter longs. And we were able to also put inside those little channels, we're able to put agarose, so some sort of nutrient inside the microfluidic chip. So this way the mycelium was still able to get some nutrients. And when the mycelium was able to slowly start colonizing the chip, we decided that we would be cutting the chip after 2 days. So once the mycelium was far enough into the chip, we would cut it and measure what's the size of initial mycelium that is inside that chip. This way, we're really able to get a very small fraction of mycelium that's inside, and be able to measure if the size do have an impact on the colonization speed on the rest inside the channel. So when we're cutting it, we're really able to see either very small, medium, or larger fragments of mycelium and measure the amount of biomass that increases over, for example, the course of 1 or 2 days, and see if smaller pieces gain much more biomass than bigger pieces. And we saw that exactly like plants, when they were too small, there was almost no growth. It's like if, for example, there's not enough cellular material to be able to start growing back. And just after that, there's just a very big peak that there's this very optimal size that very grows that made up to 10 to 15 times their biomass in just 2 days, which is absolutely crazy. And as soon as we're going to bigger and bigger sizes, it was, for example, only doubling or going up to 1.5 times their initial biomass inside the mycelium chip, inside the PDMS chip. So yeah, so we did find an optimal size in our chip, which was an initial biomass of approximately, I'd say in micrometers cube, it would be approximately 60,000, so micrometer cube. So it doesn't tell you much, but it's this is approximately the size that we're able to identify that was able to get consistently very high rates of biomass growth.
Now, I know that you used the 3D printing and you created your masters. How did you separate your final device out of the mold itself? Was there a special process that you needed in order to get the agarose off the mold itself?
So for this one particularly, we used PDMS for the mycelium chip, and no, with the 3D printer from CalWorks with the green resin, we just are able to pour our PDMS and we just remove it from the mold, and that's pretty much it. It was very easy to be able to just start with PDMS with this. So we used in the other experiments with a 2D layer with the different plugs. That's where we used agarose. We used 3D printed molds we poured agarose on top of it, and we just removed it easily, and there was not even a problem, not even an extra process. We just remove it from the mold. It was extremely easy. Yeah.
So then the agarose kind of reacted, or the process for the agarose was pretty much the same as what you've been doing for regular PDMS devices.
Exactly. It was exactly the same.
And then for the, when you test the growth of the mycelium, right, the microfluidic device, how did you, because you incorporated agar as a source of nutrients, right? So like, what did you do there? Did you kind of like insert it into the channels, or, what was the process there?
Yeah, so this is one part that took us quite some time to be able to fully get, and it was just trials and error. And what we saw is after making our full chip, so we have PDMS and we put it onto PDMS as well. After plasma bonding, we immediately put liquid agarose inside the chip, and the agarose was able to fully enter and colonize all the channels. And we just let it solidify, and that's it. The agarose was able to stay there. So since with the plasma bonding, it was really able to fully go into all the channels, and we didn't have any problems integrating the nutrients.
Okay. Did you kind of test this device with like other fabrication methods, or you went straight to 3D printing?
Straight to 3D printing, yeah. It was very easy for us. It was in the lab. We said, okay, let's try it, and we tried with 3D printing, and it's working. It was working perfectly. Yeah.
Okay.
And have you used a 3D printer before using the Microfluidics?
Yeah, that's a good question. So it was in Chris's class in my undergrad. We also had another project before that, and that's where we started using the 3D printer. But it's just the classic resin 3D printing, and yeah. I absolutely loved it. It was so fun to really be able to just have something in your mind and just take it, and it creates exactly what you need, the size that you want. And so that's why as soon as we saw we had many 3D printers that had extremely high resolution, I was extremely happy to be able to keep going and do even more of that and create even crazier devices and everything. And
so I guess the main difficulty, right, was getting those initial like small sizes of biomass, right? So I think that leads on to like your second part of your research was like, how do you continuously obtain like this very small fragment of mycelium?
Yeah, exactly. So this is because, as you mentioned, it's very good, for example, to obtain this size. But then if we want to expand, for example, for larger scale production, for example, since we need to make a lot of those very optimal sized particles to be able to make this full grown material. So how can we do that? And when we take the volume that I mentioned that didn't tell you much at the beginning, so this is basically equivalent to a sphere of 50 micron in diameter. And so when we saw that, we're like, oh, could we also adapt it and try to encapsulate this mycelium into an alginate bead? So what it is basically, can we put this mycelium particle of this optimal size, can we put it into this kind of protective shell of optimal size? And what we saw is in the literature, there's a lot of different people have been proposed to, for example, do smaller particles. And some people have seen, for example, just basically you put it into a blender. So it's kind of mechanical breaking the mycelium, and it really forms this inconsistent particle size. It damages it a lot. And there's other people that have shown that encapsulation can be done, but it's still making some very large particles because they are using external gelation of alginate, which consists of dripping the alginate. So this is the material that is going to be used to make this protective shell of around the mycelium. And we're still having some really large particles, which is not optimal for us since we need to go down much smaller in sizes for the mycelium. So there's another technique which is called it's internal gelation. I don't know why I was so it's internal gelation instead of external gelation.
Blending the mycelium. I wonder if you can actually drink that like a mycelium smoothie.
Ooh.
I think it's the new
Our-
power food.
yeah, new power food. Yeah. Okay. So they also saw that with another technique called external gelation, that you're able to also encapsulate the mycelium bead, but it kind of encapsulated as soon as you put it into this solution. So you mix it. So you mix the alginate, which is what is going to create this like kind of shell of around the mycelium, and you drip it into a solution of calcium chloride, which is basically going to immediately gel the bead. So you have really poor control over the size and the shape of the bead, which is not very what we want at larger scale, since when we want to do a larger scale, we want it to be consistent and always of the right size. And typically what is done with the dripping process. So it takes quite some time as well, and it's very hard to industrialize at larger scale. So there's other techniques that have been developed to encapsulate materials. So for example, we have a lab here as well at McGill that are encapsulating mammalian cell for type 1 diabetes. And they're using internal gelation, which instead of dripping it into the solution directly for the encapsulation, we mix it. We mix the alginate in oil, basically, and we're kind of making kind of this vinaigrette, this salad dressing. And as the more you mix this salad dressing, the more the water particles in the oil phase are going to become smaller and smaller and smaller and smaller. And this is exactly what we want to use to make those optimally sized and small enough particle for the mycelium. So we want to make them as small as possible when making your salad dressing. So it's the same thing as home. The more you shake your salad dressing, the more the water particles are getting smaller and smaller. And this is what we want to try to do to have the optimal size of mycelium in those particles.
And so, does that fully address the issue of the size? Like, do you still not get varying sizes even using that technique, or is there a fix for that?
So yeah, this is one thing that we saw, is that unfortunately it's not consistent. So even though we shake it for quite some time, the size distribution is still not optimal, and we have quite a wide distribution. So we've started using this tool, which is called the SPG emulsion, which is kind of sending this liquid through this porous membrane, so kind of like a strainer. And this way always make particles of the optimal size. So you can, for example, choose that the pore size, for example, are 50 micron, for example, which is around the optimal size of mycelium that we need. And this way can make a particle size. They're going to become just a little bit bigger, so probably around 2 to 3 times the size, so between 100 and 150 micron in size when it's going to be in the oil phase. And this way we can continuously obtain those optimally sized particles with the SPG for when we make our alginate.
So it's your vinaigrette that's being passed through the fine mesh sieve, let's say, if we make it into like kitchen terms.
So not exactly, but yes. So we're kind of making this vinaigrette. We're making this vinaigrette with the SPG system. So first we have our water phase that is going into. So our water phase, which is basically mycelium and alginate, we're passing it through this SPG membrane, and this way it would make the optimally sized particles with mycelium in it.
And this membrane that's coated around your mycelium, is it this membrane obviously is biocompatible, but is there a trigger that allows the mycelium to activate and start growing? Does it break through this membrane when it's now put into a material? Like, how does that function? Or is there this barrier? Is there any restrictions that the barrier causes for the growth of the mycelium?
Okay. So I probably just want to touch a little bit more about the SPG itself, since the SPG is just used as kind of this mesh material to make the beads. And then when the bead is out of the membrane, is just used to make the emulsion. So just to make the optimally sized, for example, alginate particle.
Okay.
But then it's going to be in oil, it's going to stir, we're going to gel the beads. So the alginate, we are going to put some acid. And this way, with the alginate in our solution, there's also some calcium carbonate that will make the alginate become stiff and become this sort of hydrogel. And this is very soft and very porous. So even, for example, when we let the mycelium culture into the beads of alginate beads, the mycelium is able to get out of the alginate very quickly. There's not a problem. It's a very porous material, and this is not a problem for the mycelium to get out of the alginate and able to grow into the material.
And have you already started testing like the seeding of the alginate beads?
So not yet. We are still developing this technique, especially with the SPG. We're into the next step. We are into this step. This is the steps that we're about to take in the next few weeks. We had some different projects that have been coming around as well that unfortunately didn't make me able to really finish this part yet. So this is the next step. So I couldn't tell you exactly what's the time frame, for example, how faster we are at the moment, if we are, but the preliminary tests are saying that it could be faster for the overall colonization. But we're not there yet, but very soon we should be able to get some very interesting results, even with the mechanical emulsion, just to test how fast we can get those materials to be able to be made.
Okay. And how long does actually like that process, like the SPG emulsion process, take? Because I know overall you're looking to reduce production time. Is this like a very complicated process where now actually you're increasing the time it takes to get to do the first stage, like substrate seeding?
So I would say,
is that not an issue?
It increases by a little bit. So for sure, you need more time. You need a little bit more resources to be able to make this. Since, the total time I'd say to be able to fully obtain the beads take me a couple hours. So it's very not that long. If we're able to save days, I think it's a very good investment to be able to make this. And when, for example, we take our substrate, our mycelium, for example, and we put it into our substrate, this is just very quick. But if we take just a couple extra hours to, for example, make optimally sized particles that can be well dispersed as well in the overall material, we think that this is a very good investment to speed up this overall colonization process.
Yeah, for sure. So then you kind of told us already, like your next step is optimizing the SPG process. Was there any other kind of research questions that you're looking to start testing in the future besides that?
Yes, so we're also thinking of doing the same experiment that we did with the plugs, but in 3 dimensions. So kind of making this transparent soil and really see the difference in, for example, the size, but in different spatial conformation. And if, for example, there's also location as well that would make an impact. So, for example, can we, for example, put it at different distances, for example, just more closer to the outside, just form the outside of the material, since it's going to form this very rigid type of barrier on the outside, and could this be enough, for example, to make our material? Since the mycelium, strangely enough, will always want to go to the air interface, the air solid interface, and form this very rigid coat. So now our question is, can we just, for example, put it just, for example, on the exterior? It's going to form the interior just a little bit, but it's mainly going to form the exterior, and if this could help us speed up the production process. This is kind of our question. Another one of our question that we're seeing right now.
Could you ever foresee that these type of beads could be created, in a factory, and then it would be shipped out? So you'd be able to transport it on site using raw products that is readily available, and then make molds at that point?
this is exactly one of our questions that we're going to be testing. So, for example, can we dehydrate, for example, those beads and ship it somewhere else, for example?
Yeah.
Could this be a good way of transporting this to, for example, other people and collaborators, if this is something that could work? Because as I mentioned, the optimal fragment size grows extremely rapidly, even, for example, in 2 days. So we would need to figure out a way to like stop it from growing. And then as soon as we put it, for example, we rehydrate them, it just starts growing. So this is one of our questions that we also want to test with those beads, for example.
Yes.
Wow.
And you plan on doing this all before you finish your master's in 6 months?
Yes, it's a very ambitious plan. But it's very fun. It's very fun. We have a lot of it's a lot of work, and we have even a lot of other projects as well going on as well. So for sure, it's going to be some busy times, but it's always so much fun. Like, for example, this is a crazy idea that came from Chris that we're testing at the moment. We did our first run. So his question was, could we make, for example, a mycelium material that had the size kind of a brain, and could we put a EEG? So it's kind of the little helmet that you put to have like kind of that measures brain waves. And can we see, for example, if the fungus is conscious? Is it responding, for example, to stimuli? And since we have a collaborator at the neuro at McGill, so the neurological department and hospital at McGill, we actually tested it. We made a brain-sized mushroom. We put the EEG on top of it and recorded for over a weekend the overall electrical activity of the mushroom. And we're planning on testing, can we, for example, test with its response, for example, to stimuli, to a lot of different other type of questions. For example, can we put it like under anesthesia? Can we put it like, for example, under chloroform and see the brain, while the brain waves, or I guess this one would be the mycelium waves, kind of just go down and go under full anesthesia.
Have you ever seen or played The Last of Us?
Yeah, I've seen the episode. Yeah, the miniseries.
If that happens, I blame you guys.
Imagine the brainwave reads out, "Get me a beer" or something. Like that.
Oh, that'd be so funny.
But that's very interesting.
Now, in terms of other biomaterials that you could make, in terms of this, the fact that you guys are using wood as the actual material that the mycelium is working with, you could use it just about any biological material that's available, correct? Like, yes. in third world countries, they use dung, buffalo dung, to build their homes. They use it as brick. So effectively, if you had some sort of mycelial beads of some sort, you could help them create actual bricks rather than the flat patties that they use for structures.
Yes. The only thing is the type of mycelium that we need to, for example, decay the wood, for example. This is the type of particular species, but I'm sure there's other species as well that can degrade other specific material. But then it gets a little bit more complicated. I think, for example, just with straw, we're seeing a little bit more problems just because the straw, it's coated with waxes, and this is very difficult for the mycelium to be able to degrade comparatively to just sawdust. So we would need some specific type of mushroom to be able to degrade, for example, other sort of material.
Yeah, it does.
Yeah, but in terms of other like construction applications, I know there's some companies that are starting to make also like a polyurethane replacement for insulation panel in your homes. For example, the blue panel that are used in your home for insulation. Well, since mycelium also has some very good insulation properties, we can also put them in walls. So as long as your wall does not get a lot of water, which I don't know if that happened to you recently in close to where you are, but in Montreal we had some really big floodings, and this would be very a big problem. For example, if all your panels would be made of mycelium, but as long as your mycelium panel does not take any water or any big humidity, I think I saw on the website of that company that it can last for up to 20 years in your house.
Following up on that, because again, when you autoclave it, essentially it can't grow anymore. If it is exposed to large amounts of water, is it possible that it does like come back live or it starts growing, or is it just it falls apart?
It falls up. Well, I think there's just going to be mold that's going to start growing on it, and then it's going to just start degrading. But I think there's some research when I went to a conference over the summer. Some people were talking about making live building, like the mycelium is just kept alive for the building that can just keep on living and use this self-regenerative characteristic to, for example, if there's a damage in the wall, it becomes it just repairs,
fuses
and fuse back together and stay and just remake this, which would be a really good alternative to other sorts of material that are not self-regenerative, where in that case the mycelium would be self-regenerative.
And would it grow mushrooms as well on the wall?
No. I don't think so, but it could be. I think they're trying to limit the fructification to not have spores going everywhere. But I'm sure it could be an interesting part, especially if you have some really rare mushroom. That could be interesting.
Solve world hunger while we're at it, Okay.
Exactly. Because the overall idea of our research is, can we try to optimize it and try to also increase the resilience of mushroom, for example, since there's a lot of problems of, for example, bringing the mushroom that is outside and bringing it into the lab, for example. And with this, for example, finding the optimal size of mushroom, could we try, for example, to find that for other types of mycelium and then be able to colonize it and bring it into the lab? Since, for example, either to cultivate, for example, rare species of mushroom, but also for antibiotics. As we know, for example, penicillin was developed from fungus. And could we try, and there's so many different fungus that we just cannot bring into the lab and study them. And this could be a very good avenue as well for this, to be able to find the optimal size and resilience of the that makes the mushroom and the mycelium resilient, that it can survive and adapt to the overall a more hostile environment, which is basically the lab, and we can start cultivating it. So this could be also another type of vision that we can envision for this sort of question and this problem that we're seeing.
And you could use any other plant that has a root system for this type of project, can you? Or is the fungus kingdom really the only type of plant, or it's not a plant, it's a species that you would be able to use?
That's a good question. I think typically the other sorts of plant are going to develop leaves and going to get a lot of nutrient from just photosynthesis in general. So I'm unsure if this could work. That would be a good question. I'm not, unfortunately, a plant biologist and would not be able to get a very good idea on this. But if, for example, we were to feed enough nutrients, for example, maybe it could help to be able to, for the plant to be able to absorb and be able to grow. That's a good question. Because
that thought crossed my mind. It was like, wow, there's so many more root plants out there. We should have tried that. But then you're right. I think the fact that the issue that you would have is that photosynthesis is the big issue, right? That's a regenerative process for other plants as opposed to a fungus. Okay.
I don't know if there is other, for example, just feeding, like, for example, some like simple sugars, for example, if it's able to be absorbed by the roots. If this could help the plants not have to necessarily do photosynthesis. So this is a great question. I think we need to ask my dad to have some more questions.
Your dad's a biologist, or?
He has an orchid greenhouse.
See?
So that's where that's where I get a lot of some of my ideas, for example, for plants and all this. Well, as you saw in the background, probably.
Yeah, I can see it. It's such a nice one there.
And yeah, but that's also as well where I got kind of this like eco mind and all this. It's really from I'd say my dad, since he also does not use any pesticides or any this. So what we're seeing right now is this is kind of a big issue. I don't know if you've seen as well, like everything that's happening with like glyphosate and all this. So it's kind of an interesting trend that we're seeing right now, and this is kind of what got me more into like kind of the sustainability and all this. And sometimes when I have some question about mushrooms, like, oh, I don't know why my mushroom is like is doing this or is doing that. It's like, oh, have you thought about this? I'm like, oh, this is interesting. So maybe my dad will have probably some insights on for example, if you just use, for example, simple sugars or something that could overcome photosynthesis that can help the plants be able to grow.
So I think that wow, that was extremely interesting and insightful, Alex. Thank you very much for joining us this afternoon. I think you gave us a very good insight into what sustainable packaging looks like in whether it's now or into the future. Thank you.
Well, thank you for inviting me. It was a very pleasure.
Likewise.
To chat as well and to meet you all. So really, I appreciate the opportunity to also talk about my research as well.
Great. And so that concludes this episode for Big Ideas at Microscale, and we will see you soon. Have a great day.